Thank you, Suman, for having me. Today we are going for the webinar in computer-aided drug design, homology modeling part. So what is the process of computer-aided drug designing? Suman has already told about the processes, these processes. So I'll be only talking about the homology modeling part. As you can see, I have circled down this homology modeling part. So why homology modeling? Development of molecular biology, rapid identification, isolation, and sequence of genes. This gives a lot of data, and this consists of different databases like NCBI, UniProt. So that database consists of different structures, sequences. So why we should do homology modeling in the context of developing country like Nepal? We do not have the resources to conduct X-ray diffraction NMR for obtaining the 3D crystal structures. So in that case, we can use homology modeling, which is a software-based approach to obtain the 3D structure of proteins. Also, similarly, we do homology modeling for that proteins that do not have 3D structures already. So in case of that condition, we should do the homology modeling. Homology modeling is basically the method that can be applied to generate reasonable models of protein structure. What does homology modeling do? It predicts the three-dimensional structure of a given protein sequence target that can be obtained from the different databases like UniProt, where we can simply obtain the sequences in FASTA format and simply input in the homology modeling server like Swiss model. It can target based on alignment to one of the more known protein structures templates. So in case of example, I'm taking the example of Swiss model. It has SMTL, Swiss model template library, where it consists of various 3D crystal structures of the proteins, similarly structured proteins. So we can align them with our target protein and create the models. If similarity between the target sequence and the template sequence is detected, structural similarity can be assumed. In generally, 30% sequence identity is required for generating useful models. So let's go on the main homology model building one of the freely available server, which can be available at swissmodel.xpagi.org. It is relatively easy than other homology model softwares. In this homology modeling workflow, you simply have to pick up the target protein sequence from the databases like UniProt in FASTA format or their ID. After taking the target protein sequence, you need to input into their server, and it will automatically align with the template library, SMTL library. So it will give the target template alignment. After obtaining the target template alignment, after obtaining more than 30% sequence similarity, we can build the models. And further models should be evaluated through QE tools. And further model evaluation should be done by other energy analysis tools. And similarly, molecular dynamics simulation. So we are not covering at the moment molecular dynamics simulation. We only how we can build a simple model. We are focusing on that at the moment. So after inputting the after sequence into the sys model server, you will get the model in PDF file format, which can be visualized by different softwares like PyMol, Chimera. So here is the model of dengue virus I've done in my thesis. So how does model will be evaluated? This is the main thing we should consider. The terms like GMQE and QMIN. Quality gives the quality estimation. GMQE means global model quality estimation, which will be ranked from 0 to 1. Higher is better. Similarly, QMIN, quality model energy analysis. It will also be in between 0 to 1. Higher will be better. This is the Swiss model obtained from that Swiss model server. Homology model of dengue virus to full length NS2B, NS3 protease. So this is all. Over to Suman.
Okay, so we have just 1 minute left for remaining portion. So if anybody has any question about this particular session, you can put on your queries. Later on, we are having another Q&A session in another session. So if anybody has any question, please. Okay. Thank you for everyone for participating our webinar. So let's have some Q&A session after this. So you can directly go to the